Hello friends, welcome to my channel. I am Rizwan Hafeez and today we are going to talk about another very important topic, that is HIV virus life cycle. We know that there are 7 important stages of HIV virus replication inside the host cell. But before talking about those stages, first of all let's talk about the structure of HIV virus and the structure of host cell which is going to be infected by this virus, so that you may get clear concept of all the stages. So first of all let me tell you that HIV virus is basically a retrovirus. Retrovirus mean the original genome of this virus will be single stranded RNA. Okay, here you can see a single stranded RNA will be the genome of this virus. But during some stage of its replication inside the host cell, this single stranded RNA will be converted into double stranded DNA. Okay, and for this process, conversion of this RNA into DNA, a very important enzyme that is called as reverse transcriptase will be present inside the HIV virus. We know that the process of conversion of DNA into RNA is called as transcription. But in this case, RNA is going to be converted into double-stranded DNA. So this is basically reverse transcription. So obviously we need reverse transcriptase enzyme, and that enzyme will be present inside the virus. Clear? Okay. Some other very important enzymes, the one is protease, is also present inside the virus, and integrase is also present inside the virus. Now, what is the function of these enzymes will be discussed in the later part of the video. Now, this HIV virus is basically enveloped virus. What do I mean by enveloped virus? There are some viruses who have extra coating other than this capsid. Most of the virus have their genome and this coating that is called as capsid. This blue color line, this blue color coating is called as capsid. Most of the enzyme have their genome, that may be RNA or DNA, and this coating that is called as capsid. But some enzymes, some viruses do have extra coating, and this coating is called as envelope. And those viruses are called as enveloped viruses, and those viruses who do not have this extra coating, they are called as non-enveloped viruses. But our HIV virus is enveloped viruses. On the envelope of this virus, two very important proteins, the one is glycoprotein or GP120 and the other one is GP41, are present. Okay, these proteins are going to attach on the receptor of the host cell. Obviously this HIV virus need to get inside the host cell for the replication, because we know that viruses, they cannot replicate without host cell. They need some host cell for the replication. So the first of all they have to attach on the host cell, and then they have to penetrate and replicate. For the attachment on the host cell, these viruses will use these protein. Okay, this GP120 will be used for the attachment, and this GP41 will be used for the penetration. Attachment and for the penetration. Attachment and penetration. Clear? Okay, now let's talk about the cells which are going to be infected uh, by HIV viruses. HIV viruses will infect our immune cells. Not all the immune cells will be infected, but there are certain types of immune cells which are called as CD4 plus cells. CD4 plus cells, like those cells which have a very important protein called as CD4 protein. This protein, this pink color protein is called as CD4 protein. Those cells which will express this CD4 protein on their surfaces will be called as CD4 plus cells, and they include uh, T helper cells, very important cell. I have talked about these cells in very detail in my immunology lectures. Then macrophages, then dendritic cells, a type of antigen presenting cells. So they are basically immune cells, and these cells express CD4 protein on their surfaces, so they are called as CD4 plus cells. So CD4 plus cells will be the target of these HIV virus. Now point is, this CD4 is a main receptor. This CD4 is a main receptor. Main receptor. But these viruses, they also need some co-receptor for the infection. And these co-receptor can be CCR5, CC chemokine receptor type 5, also called as CD195, or these co-receptor can be CXC R4 receptor, CXC chemokine receptor, are also called as CD185. So these are the core receptor. The main receptor is CD4, and the core receptor are CCR4 or CXCR4, CCR5 or CXCR4. These are the core receptor, and this is the main receptor. Now point is, in when I was explaining about the virus, I explained that this GP120 is used for the attachment. Now this is the one protein. It can attach with the main receptor, CD4 receptor. Now who is going to attach with these core receptor? This is a very interesting question and a very interesting point, because this GP is going GP41 is going to play its role in the penetration, not for the attachment. Now GP120 will attach with the CD4 cell, the main receptor. Now who will attach with this core receptor? This is very interesting question. Now let me answer this question. Once this GP120 will attach with the main receptor, that is, CD4 receptor of the CD4 plus cell, and this is HIV virus. Now once this attachment will be taken place, the conformational change in this GP120 will taken place, and its affinity for the core receptor will increase, same like this. Now after the conformational change, an additional domain for the attachment with core receptors will express. Now it will be able to attach on CCR5 or CXCR4. Okay. Now after the attachment with main receptor and the core receptor, now the first stage, that is called as attachment. Here you can see the first stage, attachment, and now attachment has been occurred. Main receptor and the core receptor has attached with the GP120, and this is the first stage. Okay. Now second stage is penetration. Now what will happen during penetration? During penetration, after this attachment, the GP41, it will penetrate inside the host cell, and it will cause the penetration or endocytosis of this HIV virus, and endocytosis will like this. Okay, endocytosis will occur. Clear? Now what will happen after the endocytosis? All the enzymes, reverse transcriptase, I have explained, reverse transcriptase, protease and integrase will also be released inside the whole cell. Now this is a CD4 plus cell. This whole cell is a CD4 plus cell, and this is a nucleus of the CD4 plus cell, and this is the virus, HIV virus. After the attachment, it has released its genome and all the enzyme inside the CD4 cell. Clear? Now this genome at this stage is a single stranded RNA. Now let me make a straight RNA for the better understanding. So this is a RNA of the virus. Now the first enzyme, that was reverse transcriptase, will be used, and with the help of reverse transcriptase a complementary strand of DNA will be added. Complementary strand of DNA will be added with the help of reverse transcriptase enzyme. Now this is RNA DNA hybrid. Now this strand is of RNA and this strand is of DNA. Okay. But the difference between RNA strand and DNA strand is just on the basis of nucleotide. Okay. This is RNA DNA hybrid. And this RNA DNA hybrid has been formed by which enzyme? Yes, reverse transcriptase enzyme. Okay. Now, after the one domain of the reverse transcriptase enzyme has RNase activity. Which activity? RNase activity. It means now the reverse transcriptase is also responsible for degrading, for destroying this RNA strand. After this, RNA DNA hybrid will be formed. Now the second domain of the reverse transcriptase will be used and this strand will be degraded. Now we have only single strand again, and this strand is of the DNA. This is the DNA strand. Now again the polymerization domain will be used and a complementary strand will be added. Complementary strand will be added, and now this a complete pure double stranded DNA will be formed. Double stranded DNA has been formed from the single stranded RNA. Clear? Now this double stranded DNA, it will get enter inside the nucleus of the CD4 plus cell or the host cell, and now the other enzyme that is called as integrase will be used. Now this integrase enzyme, it was also present inside the virus and it has released inside the host cell. This integrase enzyme will cause the integration or penetration of the virus DNA inside the host DNA. And now this virus has become latent. Latent means now it has come into a dormant state. Okay, it has become part of our DNA. The DNA, the genome of the virus has become part of our DNA. Now, the first stage was the attachment. The second stage was penetration. The third stage was reverse transcription, in which RNA has been converted into double-stranded DNA. The fourth stage is integration, in which the DNA has been integrated, has been attached, has been penetrated into the host DNA or human DNA. The next stage is replication or making copies of the HIV virus. For making copies, two stages, transcription and translation, will play a very important role. Let's see how. Now, once the transcription of this DNA will be taken place by using normal host enzymes, this gene, this DNA will also get transcribed, and from this an RNA will be formed. After the transcription, RNA will be formed. Okay. Now obviously this RNA will come out of the nucleus, and when it will attach on the ribosomes, and it will attach on the ribosome. Now this was the transcription. DNA to RNA transcription will take place, and now this RNA will come out of the nucleus through nuclear pores, and it will attach on the ribosome, and the process of translation will start. And from this translation, new proteins will be formed. Now this whole RNA, this complete RNA that is of the that was present inside the virus, now this complete RNA will get translated. So it means all the important proteins involved present in the virus will start forming. All the important proteins will start forming. This RNA will also come out of the nucleus. Some will attach on the ribosome for the translation, but some will be present as such. Okay. Now, these proteins are not mature. They are very long protein. They have to be converted into mature form, into shorter form. And for this HIV virus was also having an enzyme along with it. And what was the name? Yes, the name of that enzyme was protease. Now this enzyme will use protease enzyme to cut all these immature protein, and these protein will be made active. Now this protein will get all these protein will get activated. Now genome is there, activated proteins are there. Now these proteins and this genome will be used to assemble new viruses. Now new viruses will capsid will be formed, genome will be formed, enveloped will be formed, glycoproteins will be formed, and a full-fledged new viruses will start making inside the host cell, inside the CD4. Now this virus, it will go, it will attach with this host cell, and through the process of exocytosis it will come out of the cell. It will come out of the cell. Clear? So after the replication, the next stage was the assembly, and I have explained all these protein and the genome, they will get assembled, and a new exact copy of the HIV virus will be formed, and then budding off. Now this virus will attach with the membrane, and through the process of exocytosis it will bud out of the host cell, and it will start infecting nearby CD4 cells or other immune cells. Obviously, when there will be a virus inside the cell, when the number of viruses will increase inside the cell, some of the immune cells may get damaged, may get destroyed, and in this way our immunity start getting weaker and weaker in the case of HIV disease or AIDS. I hope you like my video and I cleared all your concept. If you like this video, please subscribe my channel, press the bell icon and share this video as well. In the next video, I will explain about the treatment options, all the medicines used for the AIDS or HIV, how these medicines target different enzymes and different proteins of this virus. Thank you so much. Take care.